So this packet is actually part of your notes. We're going to be going through the notes on this section are going to involve some lecture from me, but some discussion with you and just kind of studying some diagrams. So that's why the notes are kind of a separate thing. But these are part of your notes for this unit. So after we finish this section, or right now if you want to, you can even just staple these to your unit notes if you want to make sure that they all stay together. What we're talking about today is how prokaryotes, bacteria, regulate which of their genes are turned on and turned off. We already talked a little bit about how this happens in eukaryotes, but now we're going to talk about how bacteria turn different parts of their genes on and off. We talked about the fact already that prokaryotes don't have histone proteins, so they can't wrap up their DNA and kind of turn off sections of it in that way. So they use different types of mechanisms to control which of their genes are being expressed at any given time. So before we start, I want you to take a second and just turn and talk to somebody near you about what you think these words mean. These are words that you probably have heard in everyday life and kind of have some idea of what they mean, and you're going to hear them today as we talk about this. And the meanings in biology are similar. So turn and talk to somebody about these for a second. Next. So here are the definitions in terms of, actually, hang on. I want you to, how did I write this slide? Hang on. Promoter, operator. Okay, so this was a matchy thing. I just didn't get it that way. But you basically said these, so if we put these in biological terms, right? The promoter is where transcription starts. We've actually already used that word for the sequence of DNA in eukaryotes at the beginning of a gene where transcription starts. The terminator, you said that had to do with ending. So the terminator is the spot where transcription ends. And the operator controls it. So the operator is kind of like the on-off switch. The words induce and repress, like you said, to induce something means to start it going. To repress means to kind of stop it or slow it down. Another version of these words that you're going to hear as we talk about genes and bacteria today is something that is inducible. If inducing something means to start it, something that is inducible means it's able to be started or able to be turned on, right? And repressible means it's able to be turned off. The way that many bacteria control their gene expression is they tend to cluster genes together. And so we're going to think about this a little bit, first of all, in terms of chemical pathways. We've talked about a whole bunch of metabolic pathways, things like glycolysis and the Krebs cycle and the citric acid cycle and all of these different chemical processes that have many steps to them. Lots of metabolic processes in living things function kind of like this one that you see on the screen. You start with some kind of precursor molecule. Then there's the first chemical reaction that's catalyzed by enzyme 1 that turns it into molecule A. Then there's another reaction catalyzed by enzyme 2 that turns it into product B, and so on and so forth. So this pathway that's shown here requires 5 different enzymes to get us to the end product. There are different ways that these types of metabolic pathways can be regulated. Sometimes the end product goes back and inhibits the activity of the very first enzyme, which kind of turns off the whole thing. We talked a little bit about this. We saw an example of this in glycolysis. The end product went back and inhibited an enzyme early in the pathway. that kind of turns it off and slows it down. This is a pretty common way that eukaryotic cells especially regulate the activity of enzymes. A lot of bacteria do something a little bit different. Instead of having the end product inhibit just the first enzyme, what they do is they actually have ways to control the genes that make all of the enzymes in the pathway at once. They kind of lump all of the genes that code for all of these enzymes together, so they can turn them all on when you need this particular metabolic pathway to happen, or they can turn them all off if none of them are needed. The way they do this is by kind of clustering these genes together into groupings called operons. And that's the topic that we're talking about today, is what an operon is and of how that works in prokaryotes. So in this case, we have the X, Y, and Z gene. They can be labeled whatever the names of those genes are. But those might be maybe X codes for an enzyme that catalyzes the first reaction in a process. Y codes for the enzyme that catalyzes the second step. And Z is the enzyme that catalyzes the third. So they're all working together for the same function. In front of those structural genes, you have two sections of the operon that work to help with control. The P is the promoter region. And that's where RNA polymerase binds. And so that's where transcription begins. RNA polymerase binds right here, and then it moves that way to transcribe genes X, Y, and Z to send that messenger RNA out to be translated. The O section that's between the promoter and the actual genes, this is the operator. And like you said, the operator is kind of like the on-off switch. This is a spot on the DNA where the repressor protein can bind when it's available in the correct shape. Like any protein, the repressor protein can change shape depending on what's attached to it, okay? And like they said, if the repressor protein is attached here, it physically blocks RNA polymerase and transcription can't happen. Repressor stops transcription from happening. It represses, it blocks, it stops it from happening. The other thing that you have labeled on here is the gene I. The I gene in this picture is right next to the promoter, but it doesn't have to be. The I gene could be way back over here as well. But the I gene codes for the repressor protein. So when the I gene is transcribed and translated, it makes this protein that then goes and attaches to the operator and blocks transcription of the rest of it. So it's related to the actual operon. It's not officially part of this operon, but it's related to it because it makes that protein. So there are two main types of operons that we're going to talk about, and we'll probably just talk about the first one today. We will pick the rest of the notes on operons up after tomorrow's quiz. You do have a quiz tomorrow on the information you've learned this week. It will be through College Board. It will not be very long. It'll probably take us about 15 minutes tomorrow, and then we'll come back and kind of pick up from here. Yes, ma'am?
Is this stuff on?
I believe there is one question just to see if you know what the idea of an operon is. There's not going to be much. Good question. Okay, so you have this diagram in your note packet, and you're actually going to spend some time in just a minute here talking with some other people about this diagram. But first, I just want you to look at it and notice a couple of things. First, other than the gene that regulates the operon, how many genes are controlled within this operon? Take a look at this picture. How many genes are part of this operon right here? Hold up your fingers when you think you know how many. So I'm seeing 3s and 4s. The genes in here, so you've definitely got gene X, gene Y, gene Z. Is the terminator actually a gene? Nope, that's like the stop. That just tells it it's the end. The operator is not a gene. It's just a place where it binds. The promoter is not a gene. That's just where RNA polymerase binds. This is the regulatory gene. Is that officially part of the operon itself? No. So there are actually 3 genes that are part of the operon. There is also the regulatory gene, but that's not officially part of the operon itself. That's related, but not part of it. Second thing, when you look at this one should be pretty obvious. Where on the DNA strand does RNA polymerase bind? Locate that. See if you can figure out where RNA polymerase is in this picture. RNA polymerase binds to the promoter, right? We just said that a second ago. So that kind of teardrop shaped thing, this right here, is RNA polymerase. And just to make sure that we're clear, which direction is that RNA polymerase going to move? To the right. Yep, because it wants to transcribe these genes. In this picture right now, the operator's in the way, but RNA polymerase wants to move to the right. So what I would like you to do now, in your packet of notes, there are several questions relating to these diagrams that you can probably answer just by studying these diagrams without me telling you anything. And so I'm going to give you probably the rest of class, and then we'll pick up by discussing this a little bit more tomorrow. You can answer the question right below the diagram and the entire next page, all the way through the questions that say low lactose and high lactose, by looking at that diagram and reading that information. So you can turn to the people near you, talk about this as you go through it, and see if you can figure this out together. And we'll begin tomorrow by discussing this inducible operon a little bit more.